Okay, so Manuel apologizes he couldn't make it today, so I'll do my best to present his talk. So this falls under the big scheme, the grand scheme of looking at petroleum in the environment, and yeah, so his project is entitled MOSS, Marine Oil Spill Studies for short, and there's again a wide variety of collaborators across the GSC. And across NRCan and across DFO and other universities. So the context, the active port of Kitimat, located right here near Prince Rupert, British Columbia on the west coast, is a gateway for the export of Canada's energy resources, primarily liquid natural gas, to international markets. Current and future projects translate into dramatically increased tanker traffic in the Douglas Channel for decades to come. So that's the main goal this project is trying to address, the main subject. The objectives in particular are to examine a baseline of natural variability in the Douglas Channel, for example, temperature, productivity on a seasonal to millennial timescales, and to understand the capacity of in-situ microbial communities to mitigate accidentally released petroleum products under reduced O2 and lower pH conditions, using a series of microcosm experiments. So project overview, surface sediment and sediment trap component, known as, we'll refer to as calibration by Manuel, to assess the spatial distribution and seasonal variability in phytoplankton communities and geochemical indicators. Number two is a sediment core component, reconstructions, using short box cores right here to reconstruct past environmental variability and trace possible human impacts over the past 120 years. Using primarily lead-210 dating to obtain a good chronological model. Longer piston cores to reconstruct natural variability over a much larger time scale, the Holocene, the last 11,000 years for context. Using a C14 model to determine, and C14 to obtain an accurate age model. And what I'll be showing here is some data from microcosm experiments, lab tests, which are lab-based experiments testing the capacity of in-situ microbial communities using water and sediment to degrade petroleum products under a range of reconstructed and forecast O2 and pH conditions. So the environmental setting of this area, the Kitimat Fjord system, it's a complex fjord system on BC's northern coast. And it opens to the Pacific Ocean through the Hecate Strait, so I think it's over here. And the hydrology has a lot of small rivers, but abundant precipitation. There's a lot of natural wet, temperate rainforests in this region. And the hydrological head is the Gardner Canal, which I believe that Manuel mentioned that a lot of the high proportion of the inflow is from this water body right here, known as the Gardner Canal. And has a complex estuarine circulation system. And the surface temperature is a nice cozy 7 to 15 degrees Celsius. And its system is seasonally hypoxic. That is, it's between being aerobic or oxic and anaerobic or anoxic. And the particle flux is driven primarily by freshet and rainfall. So sediment samples, surface sediments, 37 were collected from across the estuarine system. And there's 3 years worth of sediment trap data, 3 box cores and 2 piston cores. And as it mentions here, all the material was provided by PGC and DFO. So in more detail, I'll show results from the microcosm experiments. So the first round was to examine the effect of O2 limitation on natural oil degradation rates. So to test the effect of O2 limitation on natural oil degradation rates at the sediment water interface, some material, both seawater and surface sediment from 3 stations across the estuary were collected, representing oxic, hypoxic, and anoxic conditions. And the fuel to represent potential fuel used in ships that are transporting LNG across the region. The fuel that was used was VLSFO, very low sulfur fuel oil. Methods, the bottles were incubated at 8 degrees Celsius over several months. And O2 supply was controlled from the gases in the headspace. The headspace gases were measured by gas chromatography. As is in the previous talks, the microbial communities were assessed using a 16S ribosomal RNA analysis. Oil degradation was tracked by GC mass spectrometry, and we're also going to examine carbon uptake using the C14 content of PLFAs, as was done in a similar manner to what I showed from the previous work of the Dilbit project. So there's 3 experimental setups here. There's S1, which is sediment plus water plus fuel. S2, sediment plus water, no fuel component. And S3, autoclaved sediment plus water plus fuel. So the microbes in this have in theory been eliminated and killed. So that should be, all the reactions should be inorganic and not microbial. So round 1, effective O2 limitation on natural oil degradation rates. This is the GC data from the headspace. So cumulative O2 consumption here on the left versus cumulative CO2 production. And you can see here that in the oxic and hypoxic, the anoxic cultures were barely active. There was comparable activity under oxic and hypoxic settings. And oxic cultures show the fastest rate at the start, but hypoxic cultures are more active in the end. You can see here that, within air, it's just like overtaking from the hypoxic cultures. Okay. So here's the full suite of data plus the controls. So you can see here we have O2, S1, and controls. Remember, S1 refers to the actual the microcosms with the oil, and S2 is no fuel. S3 is the kill cultures. So you can see that the O2 consumption, the background activity was not negligible in these experiments. S1 versus S2, the activity was always higher with oil across all oxygen settings. So even though there was some degradation of the background organic matter leading to O2 consumption and CO2 production, the microcosms with the oil showed more microbial activity as reflected by the O2 consumption and CO2 production. S1 versus S3, the O2 uptake in kill cultures, there's definitely, so the O2 uptake in kill cultures, but there's not O2 uptake, rather, but there's lots of CO2 released, as you can see by this slide. So one potential theory that was discussed yesterday is that maybe the autoclaving doesn't necessarily kill all the microbes, or in fact, that CO2 is being released by abiotic processes. Okay, and so here's looking at some individual compounds, looking at the effective degradation, oxic versus hypoxic. And so you can see here on the left, alkanes. You have similar degradation from various alkanes. Just looking at this qualitatively over time, you can see the degradation is similar. But for aromatics, here's an example given as methylphenanthrenes, an alkylated pH. You can see that there was more effective degradation of these compounds under oxic conditions versus hypoxic conditions. Okay, and that's all the results Manuel has to show today. And if you have any questions, please get in touch with Manuel. But I can try to answer some questions now if you have any.